Hello everyone, welcome to Brain Blitz Audios. Today in this episode of Mind Maps, we'll be dealing with the grade 12 syllabus for chemistry in CBSE. Today we're looking at the chapter Biomolecules. This is the 14th chapter in the grade 12 syllabus for chemistry in CBSE. And as always, we'll be enlisting the help of a mind map in order to get there. Mind maps are easy tools in order for last-minute preparations for various exams. However, like this one, they can be very extensive. So therefore, this video is devoted to explaining this mind map in some detail. So today, the chapter is biomolecules. So what essentially are biomolecules? The living system that is an organism is a machine which can grow, sustain, and reproduce itself. And it is remarkably made out of non-living atoms and molecules. The molecules that make up a living organism are called as biomolecules. So in this chapter, we'll be looking at the various molecules that we have, which are non-living, but they are an integral part of our system. Firstly, carbohydrates, then proteins and enzymes, then we'll be looking at vitamins, and finally, we'll be touching nucleic acids and DNA fingerprinting. So this is the standard layout for the video. And don't forget, stay back because we'll be talking a lot of important aspects about this chapter in the end. First up, we have carbohydrates. Now, for years, people thought that carbohydrates meant that they were hydrates of carbon. However, there are many molecules which are carbohydrates. That means they give us energy, but they're not hydrates of carbon. Also, acetic acid, which is a proper hydrate of carbon, is not something that gives you energy. So carbohydrates have a different definition. So carbohydrates are either optically active polyhydroxyaldehydes or ketones, or they can be classified as compounds which produce such units on hydrolysis. If you look at glucose, for example, glucose is an aldehyde with 6 carbons, so it is a carbohydrate. If you look at starch, starch is not an aldehyde or ketone. However, when you hydrolyze starch, you get glucose, fructose, etc. So therefore, you can consider starch and cellulose as carbohydrates because they give such optically active units when they are hydrolyzed. Carbohydrates are also called as by another name, and this name is saccharides. Saccharides, saccharides. And the reason why is that the term saccharide means something that is sweet because it's derived from Greek. So the earliest carbohydrates that we considered were sugars and fruit juices, etc. So the sugars such as glucose and fructose, they are carbohydrates and they taste sweet. So therefore, we considered carbohydrates as saccharides, and that's why the name still sticks today. So enough history about carbohydrates. Let's look at the classification of carbohydrates. The first kind is about the unit itself. So first up, we have monosaccharides. What are monosaccharides? Monosaccharides are carbohydrates which cannot be further hydrolyzed. So that means you cannot reduce it to further smaller units of carbohydrates. They are the basic unit of carbohydrates. Now, to give you an example, if you take starch, you can hydrolyze it to many atoms of glucose. Many molecules of glucose. But if you take one glucose molecule, you cannot hydrolyze it further without it not being a carbohydrate. So it cannot go any farther than glucose. Glucose is like a monosaccharide. So it is one of the simplest forms of carbohydrates. So if you talk about glucose, glucose is a monosaccharide because it cannot be hydrolyzed further. However, starch is not a monosaccharide because you can further hydrolyze it to other sugars and carbohydrates. Most of the monosaccharides are basically carbon compounds from 3 to 7 carbon atoms, such as triose, tetrose, pentose, etc. And they usually have aldehydes or ketones as their functional group. Monosaccharides with aldehyde group are called aldoses, and monosaccharides with ketone groups are called ketoses. Now, the most famous example of a monosaccharide is glucose. Glucose is an aldose, or which means that it has an aldehyde group, and glucose is also made up of 6 carbons, so it is a hexose. So if you have an aldehyde group and you have 6 carbons, the name that you get is aldohexose. Let's talk more on that in the next part of carbohydrates. So, glucose is one of the primary units of carbohydrates. It's a monosaccharide. So how do you produce it? The first way to prepare it is if you boil sucrose with dilute HCl or H2SO4, that means you get H plus ions, in an alcoholic solution, glucose and fructose are obtained in equal units. So as you can see, sucrose plus water, this is in an alcoholic medium, and you add dilute HCl or H2SO4, some acidic source, and you get C6H12O6, which is the formula for both glucose and fructose. Now, you might be wondering, what is the difference between glucose and fructose then? The main difference is that glucose and fructose are both what we call functional isomers. That means they have the same structure, similar structure, but their functional group is different. For glucose, the functional group is aldehyde. And for fructose, the functional group is a ketone group. So basically, you have CHO as the endpoint of a glucose molecule, but for a fructose, you have a C double bond O, a carbonyl group in the middle. And that's why glucose is an aldehyde while fructose is a ketone. And since there's a difference in functional groups, we can call these as functional isomers. That is the first way in order to prepare glucose. What is the commercial way in which we prepare glucose? Commercially, glucose is obtained by hydrolysis of starch. So you can prepare glucose from starch. What you essentially do is you boil starch. So starch, which is C6H10O5 N times, plus the required amount of water, is boiled with dilute HCl at 393 degrees Kelvin under 2 to 3 atmospheres of pressure. So there's H plus ions present from H2SO4. The temperature is 393 degrees Kelvin, and you have 2 to 3 atmospheres of pressure in that bowl. And what happens in the end? You get N molecules of C6H12O6. And you know what's the formula for C6H12O6? What's the compound? It is glucose. So you can produce glucose by boiling down starch in the presence of an acidic medium at 393 degrees Kelvin and 2 to 3 atmospheres of pressure. So now let's look at the structure of a glucose molecule. Basically, a glucose molecule, it is an aldohexose. As you can see here, the functional group is an aldehyde. You have 1, then there's 4, 1, 1, 4, and then 1. So 1 plus 4 plus 1, that is equal to 6. So you have 6 carbon and an aldehyde group. So therefore, glucose is known as an aldohexose. When we're talking about the structure of glucose, it pays to note that glucose has both cyclic structures and open structures. So the open structure is CHO, CHOH4, and CH2OH. This is the open structure. When we're talking about the cyclic structure, it has 2 types. One is alpha D glucose and the other is beta D glucose. So both have 5 carbon. So both is made up of a ring of 5 carbon and 1 oxygen. This is similar to another compound called pyran, and therefore we call these cyclic structures of glucose as pyranose structures. So glucose cyclic structures are known as pyranose structures. So therefore you can call alpha D glucose as alpha D glucopyranose, and you can call beta D glucose as beta D glucopyranose. Now remember that glucose had a functional isomer. The functional isomer that is fructose has a cyclic structure too. But for fructose, you have 4 carbon atoms in the ring and an oxygen atom to form a 5-member ring. This is similar to another compound known as furan. So therefore, the structures of fructose are known as furanose structures. So alpha D fructofuranose is one structure and beta D fructofuranose is another structure. Now remember that we gave an open structure of CHO, CHOH4, CH2OH. How did scientists find out that this was the open structure for glucose? They didn't observe glucose molecules. What they did was they took experiments on it, and then they found out that these properties satisfy the structure. So what are the properties? Property number 1, a glucose molecule, when prolongedly heated with HI, forms N-hexane. So this molecule here is known as N-hexane. And it is formed when you take glucose and you heat it for a long time in the presence of hydrogen iodide. Now, what does this prove? This proves that the 6 carbons in glucose are in a straight chain. So the first part was proven. The next property of glucose structure is proved in the second test. So what they found out is that glucose molecules can react with hydroxylamine and they form oxymes, such as this one. So this is called an oxime. And when you treat glucose with HCN, hydrogen cyanide, you get another kind of compound known as cyanohydrins. So the formation of oxymes and cyanohydrins from glucose confirmed that glucose had a carbonyl group. So basically the carbonyl group means a carbon which has a double bond with oxygen. So this is called a carbonyl group. And since glucose is an aldehyde, it has a carbonyl group. So they are correct. Now, how did they find out that this carbonyl group was aldehyde? Now, in order to find that out, what they did was they reacted glucose with bromine water. It's essentially H2O with bromine. So what they found out is that glucose, it gets oxidized to a 6-carbon carboxylic acid, this one. Now, the name of this acid is gluconic acid. So glucose gets oxidized to gluconic acid on reaction with bromine water. And the specialty with bromine water is that this is a mild oxidizing agent. So if glucose can get oxidized with mild oxidizing agents, then the carbonyl group has to be an aldehyde. And that is how we found out that glucose has an aldehydic group as its carbonyl group. Now, if you look at the structure of glucose, you see in the middle there are 4 carbon atoms, and all of them have an OH group alongside them. How did scientists know that this was present? What they did was they used a process called acetylation of glucose. So glucose was reacted with an acetic anhydride in order to form a glucose pentacetate. So this huge compound here is called glucose pentaacetate. So when treating it with anhydride, you got glucose pentaacetate. This proved to scientists that there are OH groups which are present in all of the carbon atoms, all of the different carbon atoms. So therefore, the middle structure was born. So C with an H on one end and OH on the other end. And this links with the previous carbon, that links with the next carbon. So that is how the structure is. And finally, when we oxidized glucose with nitric acid, what we got is that glucose, when oxidized with nitric acid, yields another acid, which is a dicarboxylic acid because it has two carboxylic groups. And this acid is known as saccaric acid. So the formation of saccaric acid indicated that there was a primary alcoholic group as well in glucose. As you can see in the last carbon, there is an alcoholic group, a primary alcoholic group. So by doing all these experiments, scientists concluded the correct structure for glucose, the correct open structure for glucose, which is CHO, CHOH, 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 and finally CH2OH. This is the open structure for glucose. Now, glucose and fructose are the most important types of monosaccharides. Let's look at bigger molecules, disaccharides. Disaccharides contain two monomers. Now, the monomer of a carbohydrate is a monosaccharide. So if a disaccharide has two monomers, then it has two monosaccharides. Now, how are these two monosaccharides linked together? They are linked together. They are joined by a glycosidic linkage, also called as an oxide linkage. So how do you form this oxide linkage? So they are joined, these molecules, they are joined by an oxide linkage, which is formed by the loss of one water molecule. So if you have two monosaccharides and in some arrangement you manage to take out one molecule of water, then you have formed a glycosidic linkage between the two monosaccharides. And that's how you form a disaccharide. Examples of disaccharide include sucrose and maltose. Sucrose is a combination of one glucose and one fructose, while maltose is a combination of two glucoses. And any saccharide which has three or more monosaccharides in them are termed as polysaccharides. These contain a large number of monosaccharide units which are joined by glycosidic linkages. We have three good examples for polysaccharides. Number one is starch. Starch is a polymer of alpha glucose which contains two components, amylase and amylopectin. Amylase is the water-soluble part of starch, while amylopectin is the non-water soluble part of starch. Starch's primary function is the storage of energy. So energy produced by the plants is stored in the form of starch. Next, we have cellulose. Cellulose is the most abundant organic molecule in the plant kingdom. This is basically a straight chain, and it is composed of beta D-glucose molecules. And this unique structure for cellulose makes it strong and rigid in order to support plant weight. Finally, we have glycogen. Glycogen is a branched polymer which is found in animal tissue. Glycogen is the same stuff, which is the fat. So this is, again, a storage tissue found in animals. So the so-called fat in animals is actually glycogen, and it is a branched polymer. Now, what are the importance of carbohydrates? Now, they are a major portion of our food. Carbohydrates form a major portion of our food. Most of the staple dishes around the world are made of carbohydrates. Carbohydrates are also incredibly important because they have storage molecules. So for example, starch is basically the stored form of energy. So you can use carbohydrates as storage molecules. Carbohydrates and polysaccharides, for instance, such as cellulose, they form the cell wall of bacteria and plants, which is a tribute to their strength. Carbohydrates also form raw materials for many industries, such as textiles, paper, lacquers, breweries, etc. So all of these are made up of particles, which are essentially carbohydrates. And that's why carbohydrates is very important as both our food and also raw materials for many industries such as textiles, paper, etc. Now that we've studied the first molecule, which is carbohydrates, let's look at another very important part of the food that we eat and our bodies. The second kind of biomolecule that we'll be studying is proteins. Proteins are defined as the most abundant, they are described as the most abundant molecules of any living system. So all of the machinery, such as the organs, organ systems, etc., they are actually made up of proteins mostly. Now, what exactly are proteins? Just as carbohydrates, proteins are also a kind of polymers. Now, in carbohydrates, we had monosaccharides such as glucose, which were acting as the monomer for the carbohydrate polymers. But for proteins, we have another kind of monomer. This is called an amino acid. So an amino acid is basically a carbon compound with a carboxyl group at one end and a nitro and an amine group at the other end. Then we have an R, which can be any variable carbonated group, and an H at one end. So this is the structure for a general amino acid. So amino acid, the particular thing about amino acids is that they contain amine group and also carboxyl group together. Now these monomers called amino acids are very limited. There are only 20 amino acids which are found in nature. Now among these 20, 8, these are classified into 8 and 12. 8 are essential to the humans. Essential means that we cannot produce it. It must come through our diet, while the other 12 are amino acids that we can produce. So that is how amino acids are classified. And the monomer that is amino acid, they form the structures, the complex structures that today we call proteins. Let's look at classifications of proteins. The first is on the basis of relative number of NH2 and COOH groups. Now this is pretty important because this describes the acidity or basicity of an amino acid. Now, the important thing to note here is that an amine group is generally basic in nature and a carboxyl group is generally acidic in nature. So that means if a protein has an equal number of NH2 and COOH groups, then it is a neutral protein. However, if an amino acid, if a protein has more number of NH2 than COOH groups, then you can safely say that that protein is basic. If a protein is acidic, then you can find more COOH groups than NH2 groups in that protein or amino acid. And on the basis of synthesis, you can classify them as essential and non-essential. Essential amino acids cannot be synthesized in the human body. However, Plants can synthesize it, and you can also find it in food sources. Non-essential amino acids are the other kind of amino acids which can be synthesized in the body. There are 12 amino acids which the body can synthesize, but 8 amino acids cannot be synthesized. So 12 plus 8 gives you the total number of amino acids, which is 20. Now, on the basis of shape. So on the basis of the shape of the protein that we get, there are 2 types of proteins. Number 1 is fibrous, fiber type structure. So in fibrous structure, what happens is that the polypeptide chains, they run parallel to each other. So if you have one chain going this way, another chain goes that way. So they are parallel to each other, and then they form fibers. And these fiber structures makes them quite tensile. Examples for fiber structure include silk, keratin, the stuff that forms our hair, etc. The next kind of amino acid on the basis of shape, on the protein on the basis of shape, is globular protein. Now these globular proteins are spherical. So if you have an amino acid chain, then what happens is that it starts circling like this, and over time it becomes a spherical shape to form a globular protein. Examples for globular proteins include insulin and albumin proteins. We've said that proteins are polymers of amino acids. And in carbohydrates, we've discussed how a carbohydrate, how a disaccharide and a polysaccharide are formed due to the presence of glycosidic linkages between the monomers. So what kind of linkage gives you the polymers that are proteins? So what kind of linkage is present in polymers? The kind of linkage present here is called a peptide linkage. Now, in a peptide linkage, what happens is that you form an amide bond between the carboxylic group and the amino group. So if you remember, this was NH2 and this was COOH. So what you do is you remove water from these two and you form a bond between the carboxylic group of one amino acid and the amine group of the other amino acid. And that is how the chain progresses. Now, proteins, they are formed of amino acids and they have various structures. Some of them are fibrous, some of them are globular. However, when a protein is present in its native form and you subject it to physical change, or such as heat and pressure, etc., then these globules and fibers, they unfold. So what happens is that if you have a string and then you tie it together without putting a knot into it, and then suddenly you twist it under pressure, etc., then those fibers, they come out. And that is the similar problem faced by proteins. So if you have a protein in its native form and you subject it to physical change, such as change in temperature, the helices and globules, they unwind, and the protein, it loses its structure. And when a protein loses its structure, its secondary, tertiary structures, it loses its biological activity. And that is what we call denaturation of proteins. So proteins, they cannot survive a lot of physical change. They can survive some physical change, but not all kinds of physical change, such as extreme heat. When that happens, the proteins, they start denaturing because they lose their structure. And when you lose their structure, they lose their biological activity, which makes those proteins useless. Now that was about proteins. Let's look at one specific kind of proteins, which are enzymes. Enzymes are what we refer to as biocatalysts. So these are basically globular proteins which are present specific for any particular reaction and for a particular substrate. So for example, if you have a specific reaction happening in the body, then that specific reaction has one enzyme. And suppose if this reaction has two variants with the substrate getting different for both types of reactions, then you have two enzymes catalyzing for both different kinds. The mechanism of an enzyme is that they lower the activation energy for a reaction. So when you have components, the reactants, what happens is that they form intermediates, and then these intermediates form products. So in order to form these, you need to have a specific amount of energy present within you, which is called an activation energy. What enzymes do is that they lower this activation energy to such an extent that you can do things more efficiently. You can actually, change the order of the products formed by 100 or 1,000 times when you add an enzyme to that reaction. And that's why enzymes are very effective biocatalysts. Again, enzymes face the same problem as proteins. They can get denatured because enzymes are mostly made of proteins. So when you subject them to extreme heat or change in pH, then what happens is that they lose their globular structure. And when they lose this globular structure, they lose that property for lowering the activation energy of reactants to form products. Now, we were talking about macromolecules such as carbohydrates and proteins. Now, there are other essential things that the body requires, but they do not require them in a large amount. Meet the vitamins, organic compounds which are required in diet in small amounts to perform specific biological functions for maintaining the body and for its growth. Vitamins are so important that deficiency of vitamins, they cause diseases. For example, a deficiency of vitamin A gives you night blindness, and a deficiency of vitamin C gives you scurvy, and a deficiency of vitamin B gives you beriberi, and so on. So vitamins are very important. You do not require them in large amounts, but you need to have them in your diet. Otherwise, you're going to have to face extreme problems. The problem with vitamins is that they cannot be synthesized by humans. So us humans, we can't synthesize vitamins. So the only other way to get vitamins is through the diet. And fortunately, these vitamins are present in plants. So therefore, you can eat plants in order to get those vitamins. Again, specific plants for that matter. Now, when you're classifying vitamins, vitamins are classified into two types, fat-soluble and water-soluble. Fat-soluble vitamins are the ones that are soluble in fats and oils, but you cannot dissolve them in water. Water-soluble vitamins are those which can be dissolved in water. Now, when we're talking about nomenclature in vitamins, we use letters in order to differentiate each vitamin. So for example, the fat-soluble vitamins are vitamins A, D, E, and K. And the water-soluble vitamins are vitamins B and C. Now, if you see here, it's written B group because under vitamin B, you have many types such as B1, B3, B12, etc. So you have a B group and C. An important point to note here is that water-soluble vitamins, such as vitamins B and C, can be lost through the urine. Because since they are water-soluble, they can pass through urine if they're present in large amounts. So it's important to take them in small quantities. Fat-soluble vitamins are the ones which dissolve in fat, so they do not have this problem. But they are also very important in order to maintain our healthy existence. Now, we know that carbohydrates here gives you energy and forms the structure for plants. Proteins, they form the structures of every cell and most animal tissues. Vitamins are important for metabolism. Enzymes, they catalyze the body reactions and they help you survive. But for all of these to exist, you need some genetic code. You need some molecule which can translate itself to form these proteins, carbohydrates, etc. So what are these kind of molecules? These are the molecules that we call nucleic acids. Nucleic acids, they are basically called polynucleotides. So these are made up of nitrogenous bases, a sugar, and a phosphate. And when you clump nucleic acids together, the largest of these units, they're called chromosomes. Chromosomes are the particles that we see in the nucleus responsible for the heredity of us humans, animals, etc. The chromosomes are made up of proteins, the histone packaging proteins, and the other kind of molecule that we call nucleic acid. Nucleic acids today are divided into two types: deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. The DNA is the molecule with the famous double helix structure, and RNA is a molecule with mostly single helix with a single strand. So DNA contains two strands connected by nitrogenous bases, while RNA is just basically one strand with nitrogenous bases jutting out through one side. Now, when we're talking about composition of nucleic acids, DNA has a different kind of sugar than RNA, and that's why they have their differences. The sugar in DNA is beta-2-deoxyribose. Ribose is a 5-carbon sugar. And when the ribose molecule is missing an OH group, it is called deoxyribose. In RNA, the OH group is present, so therefore it is called ribose. Another difference between DNA and RNA occurs in the nitrogenous bases that are present. So in nature and in nucleic acids, you basically have 4 nitrogenous bases: adenine, guanine, cytosine, and thymine/uracil. So the difference is in this part. We said that in nucleic acid, there are basically 4 types of nitrogenous bases. For DNA, the 4 are adenine, guanine, cytosine, and thymine. But for RNA, it is adenine, guanine, cytosine, and uracil. So uracil is basically thymine without a methyl group. So there is a difference of a methyl group between thymine and uracil. So uracil is the one which is present in RNA, and thymine is the nitrogenous base which is present in DNA. So suppose you have a nitrogenous base. How do you form it to a DNA? First, the nitrogenous bases, they are attached to a carbon of sugar, basically the first carbon. So base plus sugar gives you an arrangement known as a nucleoside. Now, these nucleosides are further bonded with each other by phosphate bonds. So nucleotides are formed by link to phosphoric acid at 5' of sugar. So the 5th carbon of sugar contains a molecule of phosphoric acid to form a nucleotide. And when you arrange these nucleotides in specific orders, you get polynucleotides, which are what we call nucleic acid, the polymers. So you have base jutting out of one end, sugar is bonded to phosphate, then it's bonded to another sugar, which is bonded to a phosphate, sugar, phosphate, etc. It goes on that order. And the nitrogenous bases, which jut out to the sides, they also form hydrogen bonds with the complementary nitrogenous bases at the other end. adenine is complementary with thymine, and guanine is complementary with cytosine. So for RNA, instead of thymine, it'll be uracil. So adenine forms 2 hydrogen bonds with thymine or uracil, and guanine forms 3 hydrogen bonds with cytosine. So these are intermolecular hydrogen bonds, which hold the 2 strands together. Now, what are the biological functions of this kind of molecule, the nucleic acids? Nucleic acids are the chemical basis of heredity. So all of the code, all of the information that is required to create an organism is present in nucleic acids. These are responsible for the identity of different species of organisms. Different species have different kinds of DNA, even though the change is very little between species. Nucleic acids are responsible for protein synthesis in a cell. So if you learn the chapter molecular basis of inheritance, then you would know that the DNA in a human is converted into RNA, and that RNA basically codes for more protein. So the protein in our body is essentially the work of nucleic acids getting coded. So when you code a nucleic acid, you form proteins by stacking up various amino acids together. So that is why DNA and RNA are very important. Now, we've often heard of fingerprinting in order to identify a person. You ask for the person's fingerprint, and then you try to find out if it's the right way to go, if he's the right person. Now, what is DNA fingerprinting? In DNA fingerprinting, what we essentially do is we find out unique sequences of nitrogenous bases, variable number of tandem repeats, etc., on a person's DNA. So basically, we're taking the DNA and finding out the most important parts of it that differentiate each person. So after finding that out, we compare it with DNA found in areas in order to mix and match to find out whether it was the same person who was present there. DNA fingerprinting is very essential in forensics, and you can also find it being used in accidents in order to find out who the deceased person was. Now, if you have parental disputes in your family, chances are that the lawyers would also ask you to take a DNA fingerprinting in order to find out the ancestry. And when we're talking about evolution, people use DNA fingerprints in order to find out our ancestors. So they take up this DNA fingerprint and then they compare it with ancient organisms in order to find out where our DNA came from. So that helps us out in evolution. So that is how nucleic acids and DNA fingerprinting are essential parts of our body. That ends this unit, biomolecules. So this chapter, we've looked at the energy source for us, that is the carbohydrates. We've also looked at the building blocks of our machinery, that is proteins, the regulators for metabolism, the vitamins, the catalysts, which are enzymes, and finally, the coders, the genetic code, which is found in nucleic acids, and how we can identify people using nucleic acid code. So we hope you found this episode interesting. For more of our videos on various mind maps, don't forget to click the link in the description down below, which leads you to more mind maps. To be a part of our channel, don't forget to subscribe to it. The name is Brain Blitz Audios. So until the next webisode, take care, stay safe. Bye-bye for now.